You do not develop a brain in isolation. The human brain develops inside connection. To understand how social isolation affects the brain, it helps to look at how the nervous system formed in the first place. For almost all of human existence, survival unfolded in groups. Food gathering required coordination. Protection relied on shared vigilance. Learning happened through observation and imitation. Emotional regulation emerged through proximity and feedback. Nervous systems adapted to this environment by treating social input as a primary organizing force. Faces became anchors of attention. Voices calibrated emotional states. Shared timing structured perception. Memory formed around collective experience rather than isolated events. When these conditions change, the effects often appear as social isolation, brain fog, and memory problems. Thinking slows. Recall weakens. Experiences stop settling. This pattern reflects the neuroscience of loneliness and cognitive decline, a process shaped by how connection stabilizes neural systems over time. Connection shaped cognitive stability. This stability depended on continuous social feedback. As long as these signals were present, neural systems aligned efficiently. When they weakened, internal priorities shifted. The human brain evolved under conditions of constant interpersonal regulation. In modern environments, these conditions changed. Over time, daily life moved toward reduced physical presence. Living arrangements shifted. Work migrated indoors and online. Communication increasingly relied on mediated formats. Large-scale disruptions further limited routine interaction. These changes altered the frequency and quality of social input reaching your nervous system. You may recognize this shift in your own life. Fewer spontaneous conversations. Fewer shared meals. Fewer moments of unplanned presence. More screens. More silence. More days passing without real interpersonal calibration. At first, the consequences appeared most clearly among older adults. Individuals living alone, separated from family networks, or experiencing reduced mobility, showed accelerated cognitive decline. Memory retrieval slowed. Orientation weakened. Emotional recall lost detail. Longitudinal observations linked prolonged isolation to increased risk of neurodegenerative conditions, including Alzheimer's disease. Later, similar patterns emerged across younger populations. You may have noticed this yourself. Thoughts moving more slowly. Words sitting just out of reach. Mental fatigue appearing despite little physical exertion. Sitting down to start a task and feeling drained before you begin. Many people describe this as a diffuse alteration in mental clarity. This experience became widely known as brain fog. The mechanism underlying this phenomenon resides in neural connectivity. Your brain functions as a distributed network. Billions of neurons communicate through pathways strengthened by repeated activation. Frequent signaling stabilizes connections. Reduced activation loosens them. This principle governs perception, movement, emotion, and memory alike. Connectivity follows use. When you interact regularly with others, multiple systems activate at the same time. Visual recognition, auditory processing, emotional interpretation, predictive modeling, and motor coordination operate together during shared presence. This integrated activation maintains efficiency across networks. Think of a simple conversation. You read facial expressions. You adjust your tone. You sense emotional nuance. You anticipate responses. All of this happens simultaneously. When interaction decreases, integrated activation declines. The effect often appears first as reduced processing speed and attentional stability. Following conversations feels more effortful. Complex ideas require more energy. Over time, structural changes follow functional shifts. Social cognition relies on complex coordination. It includes recognizing faces, interpreting emotional expressions, decoding tone, tracking intentions, and anticipating behavior. It also includes the ability to understand that other people have their own desires, goals, and intentions. This capacity is known as theory of mind. You use it constantly. When you sense irritation without words. When you realize a reaction is not really about you. When you adjust your behavior because you can infer what someone else might be thinking or feeling. These abilities develop early in life and remain plastic throughout adulthood. Their maintenance depends on continuous engagement. When activation drops, precision erodes. Functional change precedes anatomical change. Large-scale neuroimaging studies, analyzing structural MRI data from 32,000 participants, identified consistent patterns associated with prolonged social isolation. Reduced volume appeared in temporal regions, involved in language and recognition. Frontal areas associated with integration, and planning showed altered density. The hippocampus, essential for emotional memory and contextual binding, displayed measurable shrinkage. The hippocampus plays a central role in emotional memory. Reduced hippocampal volume has also been observed in depressive states. These regions overlap with networks affected by chronic stress, inflammation, and prolonged emotional strain. Memory depends on consolidation. Experiences require emotional salience and contextual embedding to stabilize into long-term storage. Social interaction provides both. Shared presence anchors events through feedback, narrative structure, and emotional resonance. You may notice this when you look back on recent weeks. Days were busy. Tasks were completed. Appointments happened. Yet little feels memorable. Conversations blur together. Time compresses. When interaction weakens, experiences pass through awareness without anchoring. Days remain full. Schedules remain dense. Recall fragments. Time loses depth. This pattern reflects altered encoding rather than lack of experience. Your nervous system interprets isolation through an evolutionary framework. Across ancestral environments, separation from the group increased vulnerability. Reduced access to shared vigilance elevated risk. Neural systems adapted by reallocating resources toward threat monitoring and energy conservation. This reallocation narrows cognitive bandwidth. Attention shifts towards scanning rather than integration. Emotional range contracts. Exploratory behavior decreases. Memory encoding weakens. You may experience this as low-level tension, increased irritability, or a persistent sense of unease without a clear source. These shifts occur automatically and gradually. At the individual level, they feel subtle. At scale, they become visible patterns. Despite these changes, neural systems retain adaptability. Your brain maintains the capacity to reorganize connections across the lifespan. This capacity, known as neuroplasticity, allows weakened pathways to regain strength when activation resumes. Recovery depends on accumulated flexibility. Cognitive reserve functions as a buffering capacity. It reflects your brain's ability to compensate for structural loss through alternative pathways and distributed processing. Individuals with higher reserve maintain function despite reduced volume or connectivity. You see this difference when some people remain cognitively sharp into their 80s, while others struggle decades earlier. Reserve accumulates through sustained engagement. Formal education contributes by training prolonged attention, abstraction, and symbolic reasoning. The effect depends on learning itself rather than institutional prestige. Occupational complexity adds another layer. Tasks requiring problem-solving, integration, and autonomous decision-making stimulate distributed networks. These differ from purely stressful work. High workload and unrealistic deadlines generate strain without strengthening cognitive reserve. You recognize the difference. A job that only exhausts you drains energy. A demanding task that requires thinking feels effortful, yet coherent. Mental activities outside formal work reinforce these systems. Reading, writing, puzzles, language learning, and musical training activate overlapping circuits. Physical movement supports neural growth. Regular activity increases signaling molecules associated with synaptic formation, strengthening communication across regions. Social interaction amplifies all other inputs. Conversation engages language, emotion, memory, prediction, and motor coordination simultaneously. Shared presence provides multisensory feedback unavailable through solitary activity. Nutrition, sleep, and emotional regulation shape baseline capacity by influencing inflammation, hormones, and energy availability. Together, these inputs sustain neural resilience. Prolonged isolation reduces several of them at once. Movement decreases, learning narrows, interaction thins, stress accumulates. The cumulative effect reshapes neural architecture. Recovery follows activation patterns. When interaction returns, dormant circuits respond. When learning resumes, connectivity strengthens. When movement increases, signaling accelerates. Over time, coherence rebuilds. This process unfolds gradually,